All right, so now I'd like to introduce Tricia Tamsen from who's a single cell spatial lab manager of the Advanced Genomics Core and the BRCF Advanced Genomics Core, the Rogel Cancer Center Single Cell Spatial Analysis Shared Resource. That's a mouthful. Thanks, Tricia.
All right, good morning, everybody. So I am Tricia Tamsen. I'm the single cell spatial lab manager at the Advanced Genomics Core, and I'm here to talk about our single cell offerings this morning. So when we think of single cell, we consider three broad approaches. There is the droplet-based version, things like DropSeq or 10x Genomics, the split pooling, so you're talking about Parse Biosciences or Scale Biosciences, and then direct cell lysis. So everything has pros and cons. It's entirely dependent on your experimental goals, what you're looking for. You'll notice that there is an asterisk on the size limitation on the droplet-based. We only run 10x Genomics microfluidics in our lab, and 10x just came out with new chemistry that they claim makes it any size cell is available now. We haven't tested it, but that's what they're claiming. So opens some doors if that's true. And then direct cell lysis, I'm not going to talk about later in this presentation at all. Just know that we can do this. This is based on picking individual cells by needles or pipettes or flow cytometry and dispensing it into a 96 well plate. So whatever kind of single cell you're talking about, your sample prep is the most important part. And that's because you want high quality, low debris. You want it to be alive. Because otherwise, well, all of your genes are ApoE or mitochondrial, and that's not interesting. But we really need debris-free, because otherwise you're going to get lots of background, and now you can't call cells versus background. That's a problem. Let's confound your analysis with that one. And then we're always looking for a high number of cells. A lot of these approaches rely on a certain number of cells dropping out during the process. And so we need to start with quite a few cells, quite honestly. Some things to keep in mind with your sample prep. Sorry, Dave, but y'all in flow overestimate your cell counts by 30 to 60%. By the time they get to the core, no, you do not have 100,000 cells. You maybe have 50,000 cells. So we always say sort as many cells as you can so that you have enough, provided that some are going to drop out there. There is a dye, it's called Ruby, that we've seen people use that does do a really nice job of separating cells from debris. And if you have a small population of cells, it has to be spun down, when you get done with flow, because we need a high concentration, and just stick it in 30 microliters of buffer, because we can dilute so much easier than anybody can try and spin down and concentrate and lose more cells in the process. And any media pretty much is acceptable if you're dropping off for fresh cells, because DMEM, RPMI, I don't really care. I care that you don't have more than 10% FBS or 2% BSA and no EDTA in your buffer. Whatever is going to keep your cells happy is what we want to see. So the 10x Genomics suite of offerings: RNA, TCR and BCR profiling, proteomics with cell surface proteins, epigenomics by including ATAC-seq in there. We can do CRISPR screening. We can do long read sequencing by taking the cDNA library that we get from your single cells and running them on our long read Oxford nanopore system. All options, all available. Every time 10x Genomics runs something new, we're running it within a week or two. So whatever kind of analysis you guys are looking for, we can probably help. And living cells are hard. I don't know how many of you are actually on North Campus, but the Genomics core is. And it can take you an hour to try and get the research shuttle to come from other parts of campus. So your sample is dying while it's sitting on ice. The transcriptional activity is changing. The transcriptional activity probably changed while you flow sorted. So these are all some constraints that can be overcome with a fixed system. So 10x now has this single cell RNA flex system. It starts with a fixation. You lock in your cell state as fast as possible. And instead of doing a poly-A capture of your transcripts, we're actually going to use probes. We'll hybridize the probes to your fixed cells before running it through the chip and continuing on with your analysis. It is still compatible with cell surface proteins. And this is how these probes work. So there is a left-hand probe and a right-hand probe. They have to sit down and be ligated together in order for the transcripts to actually be captured. And then they have the capture sequence on the very end so that they're pulled by the gel bead in the 10x system. It does do threefold coverage of most genes. So 3 different parts along the transcript are assayed, except for some of your really high expressors. I don't think anybody wants to waste their sequencing time on mitochondrial RNAs, unless that's what you're really interested in. But when it takes up 30% of your sequencing depth, let's not do that. Let's spend our time trying to find the more, the less expressed genes. This tends to be more sensitive because you're profiling the genes in 3 parts. You get to find some of those really low expressed genes. So there's a full set of coding genes. You don't get TCR, you don't get HLA or some of those things. But most importantly on these probes is that gray probe barcode on the right-hand side, because what that's going to let you do is multiplex. You're multiplexing at a transcriptional level. So when we hybridize the probes, say you have 4 samples in your set, we're going to use a probe barcode set for each sample. 1 gets 1, 2, 3, etc. And we pull them together. So now you have 1 reaction, and it contains 4 samples that can be parsed out at the transcriptional level at the end. So you get a really, really clean data set. I don't know if any of you ever did Cellplex. Don't do that. 10x has discontinued it. But even the antibody labeling that people have traditionally used for multiplexing can be very dirty because you're labeling a cell membrane. You're not labeling the transcripts. And so it's just dirty. It's dirty. You lose a lot of cells because it can't distinguish the cells from the background. And this can. The data is just fantastic. The Flex system also lets you start with pretty much anything. Whether it's a cell line or a cell suspension, whether it's fresh or frozen tissue. We even take FFPE scrolls and we can give you single cell data from that tissue. We isolate a combination of cells and nuclei, whether that's from the frozen tissue or the FFPE block. And it's just amazing how you're able to get the single cell data from these challenging cell types. So this is just some flex versus fresh things to think about. You know, as long as all you're looking for is gene expression of a sample, flex is the way to go. We have quite a few people who said, you know what, yes, I am just looking for gene expression. I'm going to run this pilot. And we're like, well, for, I don't know, instead of a $5,000 experiment, how about you run a $7,000 experiment and you get pretty much your whole experiment done in 1 sample set, because the flex does bring our cost down to about $750 a sample, as compared to the library prep for a fresh sample is $1,900 a sample. So it's also really great if you have something infectious. We'd love for you to fix that. There are probes that can be spiked in so that you can find that virus, find the whatever you're looking for later. And, you know, obviously that's one limitation. You can't go back to your data later and say, but I wanted to find my virus, and you can't find it now. I can't go back and find your GFP for you. That has to be spiked in the front end. But, you know, as long as you've done that pre-planning, it's a really great system. So changing gears a little bit to talk about the split-seq options. So split-seq actually uses the cell itself as the partition. So cells are still fixed and permeabilized, and then barcodes are successively put onto the transcripts through multiple rounds of splitting your cells out, doing a barcoding round, pull them back together, split them out, pull them back together. So that after multiple rounds of pooling, what you're looking at is this in the middle, where you can say, well, genes A, B, and C clearly go to cell 1 because they have the same barcodes on each transcript. So that we have two different options, Parse Biosciences or Scale Biosciences. Both are relatively similar. It's just kind of the add-ons and what you're looking for. The scale methylation is not actually run by us. We actually run that through Claudia at the Epigenomics core, but she's in the process of onboarding that, and we can support the sequencing still on the back end. And that's pretty much all I had. So this is how to get in touch with us and come talk to us about your experiment. We're happy to help. And let me know if you have any questions.
The last example, can you clarify which stage is the last stage of the single cell stage before we use the fix?
You can do any of it. So we can take your fresh cells, we can take your frozen tissue, we can take your FFPE scroll, and we'll do it all. We'll fix it, we'll dissociate it, we'll do whatever needs to be done. Drop it off wherever you're comfortable with.
Or the instead of flex is better.
It's cleaner. It's cleaner and it's cheaper. So as long as all you're looking for is the gene expression, you aren't looking for your VDJs, you aren't looking for any of that. It's an amazing system.
We would allow to learn that where, which step.
Yep. Any of the steps, we can do them all. Yes.
Here, wait a minute.
Oh.
The microphone.
Yeah, it's a very beautiful presentation. I have one question, as the sir explained about the sample preparation is the key. So like we have cells which are isolated from tissues, and this is a heterogeneous population. So facts cannot be applied because we want to evaluate the whole heterogeneous population, what cells are important, for that particular regenerative purpose. So we can also get that heterogeneous population directly, right?
Yes, absolutely.